Hello students, am I audible? So today's session we will be having a new topic from the chapter biomolecules. So one of the very important biomolecule present in all living body, that is the nucleotide. Can anybody say what do you mean by nucleotide, explanation of nucleotides, or many nucleotides that join to form what? Yes. What is that? Nucleic acid. Okay, so what are nucleic acids? Nucleic acids are the fundamental genetic material of all living organisms. When you check all the living organism, the genetic material present inside the cell which control all the body activities, that is through the nucleic acids. There are two types of nucleic acid. One is called a DNA and the second type is RNA. Most of the living organism, the genetic material is DNA. But some organisms we can see the genetic material as the RNA. Can anybody say the difference between the full form of DNA and RNA? Yes, DNA is deoxyribonucleic acid and RNA is the ribonucleic acid. So in the nucleotide, why I am using this nucleic acid topic here? Because nucleotides are the monomer which join to form the polymer is called nucleic acids. So nucleic acids are basically biomacromolecules. Biomacromolecules. Macromolecule means the larger molecules, right? And these macromolecules are made up of the micromolecules or the unit called what is that? It is called a nucleotide. Okay, so today's session we will be studying properly the structure of nucleotide, the components of nucleotide. So after the session you will be thorough with the topic nucleotides and we will be discussing the important questions from the topic. Right? So let's start with the nucleotide. So nucleotides are the basic building block of nucleic acids. And these nucleotides are made up of three components. The very first is the nitrogenous base, second pentose sugar, and the third one is the phosphate group. These three components join together to form a single monomer called nucleotides. Right. So once again, what are the components of nucleotides? There will be a nitrogen base plus pentose sugar and a phosphate group that together form the structure called a nucleotide. So what is nitrogen bases? Nitrogen bases in nucleic acids or the nucleotides that consist of two different types. Okay, so nitrogen bases are broadly classified into two categories. One is purines, they have a double ring, and another one is the pyrimidines, they have only single ring. Okay, so remember nitrogen bases are classified into two types. They are the purines and pyrimidines. Purines are of two types, adenine and guanine. Remember adenine, guanine. And when you check both the nucleic acids in RNA and DNA, both in RNA and DNA, adenine and guanine present. Remember, adenine found both in DNA and RNA, also guanine also present both in DNA and RNA. Coming to the pyrimidines, there are three different types of pyrimidines there. Cytosine, thymine and uracil. So cytosine found in both DNA and RNA. Thymine found only in DNA. Uracil found only in the RNA. So this session is very, very important. You can expect one question from this part. How it comes? The question will be like, which of the following are not present in RNA? So, or which of the nitrogen bases present only in DNA? Like that question comes. So adenine, guanine. So which are the nitrogen bases which are common for both the DNA and RNA? Adenine is there, guanine is there, and cytosine. These three are present in both the nucleic acids, RNA and DNA. But thymine is present. You have to check the spelling of thymine, Thymine. There is one more, is there thiamine, Thiamin. That is different. It is a vitamin B complex. Here the spelling is thymine, Thymine. So thymine found only in DNA, very, very important, okay? And then uracil is the nitrogen base which is present only in the RNA. So sometime we will be having confusion with these purines and pyrimidines, remembering, okay? So purines, we can easily remember cut, C-U-T, cut, okay? C for cytosine, U for uracil, and T for thymine. Is that clear? The classification of nitrogen bases. Nitrogen bases are one of the component in nucleotides. And there are two types of nucleotides, purines and pyrimidines. Purines are of two types, adenine and guanine. And pyrimidines, three different types are there, cytosine, uracil, and thymine. Okay, so this is the structure of different bases. Adenine, guanine, they have a double ring. You can see 9-membered double ring there. These two, okay. But here a 6-membered single ring is there in the case of pyrimidines, thymine, cytosine, and uracil. They have a 6-membered, that single ring is there. Okay, so there will be some changes in the position of the nitrogen in the case of adenine, guanine, thymine, cytosine, and uracil. And base pairing rule is another important part. Base pairing rule. Okay, so when there is nucleic acids formation time, especially in the DNA consists of two strands. Okay, this is one polynucleotide chain, there is another polynucleotide chain. So when these two polynucleotide chains join, there will be that will be joined in the base pairs, in between the bases only. So the base pairing is one important part here. You have to remember, always remember, adenine always pairs with the thymine and vice versa. Guanine always pairs with the cytosine and vice versa. Okay, so in the case of DNA, adenine and thymine, you can see here two lines in between the adenine and the thymine. Here three lines are there. What does it indicate? Yeah, two lines. This one is the hydrogen bond. Okay, so in between the nitrogen base, there is hydrogen bond formation is there. Hydrogen bonds are there. Okay, so in between here two hydrogen bonds. So adenine always pair with the thymine, adenine thymine, with the help of two hydrogen bond, and guanine cytosine, in between there are three hydrogen bonds there. Okay, so when coming to the nucleic acid RNA base pairing, adenine, there is no thymine. So instead of thymine, what is there? Thymine is replaced by uracil here. So adenine binds with or pairs with the uracil with the help of two hydrogen bonds. Guanine pairs with the cytosine with the help of three hydrogen bond. Otherwise, vice versa. Cytosine to guanine, uracil to adenine. Okay, so two hydrogen bond, three hydrogen bond. Remember. And you can see from here, adenine is what type of nitrogen base? It is the purines, right? And thymine is pyrimidine. So always remember, see here also, if you check a guanine is purines and cytosine is the pyrimidines. Then here also adenine, purines, pyrimidines. Purines, pyrimidines. Always remember, purine pairs with the pyrimidines, not like a purine. Okay, adenine and guanine will not pair. Only adenine, guanine with the cytosine, or cytosine with the guanine. Then adenine with the thymine, or adenine with the uracil, like this only. Okay. So there will be triple bond you can see in between the guanine and the cytosine that gives more stability. So more stable means that structure is very good for as the genetic information for transcription, replication and all. Is that clear about the base pairing? Moving on to next, second component is the pentose sugar. Pentose means 5, right? So pentose sugar consists of 5 carbon atom. So in the nucleotide, you can see two different types of pentose sugar. One is called a deoxyribose sugar and another one is the ribose sugar. Deoxyribose sugar makes the DNA and ribose sugar makes the RNA. So what will be the difference between DNA and RNA? Both are 5 carbon sugar. So when you check the structure of the ribose sugar, so how will you draw the ribose sugar? It is in the furanose form, okay? Like this, pentose sugar, the first carbon atom, second carbon atom, third, fourth and the fifth there will be CH2OH. This is a basic structure, right? So in the second position there is H and OH in the case of ribose sugar. Okay, but in deoxyribose sugar, deoxy, removal of the oxygen. Okay, so in the deoxyribose sugar, in the second carbon atom, oxygen is not there. Missing of oxygen from the hydroxyl group that makes the DNA more stable. And it is good for the as a genetic material. Okay, so genetic material DNA, if it is more stable, that is good for the one of the important characteristics of genetic material, right? So remember, ribose sugar, they have in the second carbon atom H and OH is there, but in the case of deoxyribose sugar, in the second position, second carbon atom, oxygen will be removed. So that is one of the structural difference between the DNA and the RNA. So structural difference between the ribose sugar and the deoxyribose explain why DNA serves as the stable genetic storage while the RNA function as a dynamic cellular process. Note down this point, all of you. Anybody, any doubt is there from this part? Okay, so we have learned the components of nucleotides. The first is the nitrogen base, second is pentose sugar, and the third component is the phosphate group. Phosphate group in nucleotide is an acid called, it is an acid called a phosphoric acid. Phosphoric acid. H3PO4. Phosphoric acid. So because of the presence of this acid, phosphoric acid only, the nucleic acids, they are negatively charged. Note down this point, they are negatively charged or it is acidic in nature. 2 points, okay? It is negatively charged because it is acidic in nature, and acidic nature is due to the presence of the phosphate group. Why? Because the phosphate group here is the phosphoric acid. And this phosphate group has a critical functions. It acts like an energy storage. Okay, energy storage, we know adenosine triphosphate. So there the adenosine triphosphate, it is a high energy bond storage is there inside. And chain formation like a phosphodiester linkage, when we are discussing more detail, then we will discuss that what is that diester bond, a phosphodiester bond. And this, because of the phosphate group only, DNA got that negative charge, and this negative charge prevent the DNA strands from collapsing. So these are the significance of phosphate group in nucleic acids. So what are the components of nucleotides? Pentose sugar is there, nitrogen base and a phosphate group. Then there is a term nucleoside. You can expect the question from this topic. What is the difference or distinguish between nucleoside and a nucleotide? We know that a nucleotide consists of three components: base, sugar, nitrogen base is there, the sugar is there, and a phosphate. But nucleotide without phosphate group is called a nucleoside. That means only the nitrogen base is there. Okay, so nitrogen base and the sugar, whether it is ribose sugar or deoxyribose sugar, that form the nucleoside. There is no phosphate group. So it is electrically neutral. When the phosphate group is added to it, then that gives a negative charge. You can see here in the picture here, the nucleotide and the nucleoside. Okay, so let's study the structure of the how to make the nucleotide. I have told you there is a furanose form of the ring form. There are two ring forms are there in the case of carbohydrate. The sugar one is the pyranose form and another one is a furanose form. The pendo sugar, you can see the furanose form. Okay, so this is first carbon atom, second carbon atom, third, fourth and fifth carbon atom. Here CH2OH there. So in the first position only what is attached, the nitrogen base will be attached in the first carbon atom. And for attachment or binding with the nitrogen base, there should be a bond. Okay, so what is the name of the bond? This is called a N-glycosidic bond. N-glycosidic bond. Right? So this sugar, whether it is a ribose sugar or a deoxyribose sugar plus nitrogen base when bind with the help of glycosidic bond, the whole structure is called a nucleoside. In this nucleoside, in the fifth position of the carbon atom, when the phosphate group is attached with the help of ester bond. Okay, so remember ester bond. What type of, since this is a phosphate group, so it is called a phosphodiester bond. What is an ester? See here the OH group is there in the CH2OH and this is the acid, phosphoric acid. So the bond formed between the alcoholic group and the acid. So this bond is called a ester bond. Since this is a phosphate group, the name is phosphodiester bond. So how many bonds are there in the nucleotide? The whole structure is the nucleotide. So in this whole structure, nucleotide, how many bonds are there? You can see two bonds, right? One is the N-glycosidic bond formed between the nitrogen base and the sugar. Another one is the phosphodiester bond that is formed between the fifth carbon atom CH2OH, okay, OH group and the sugar and OH group from the sugar and phosphate group, that is phosphoric acid. Okay, so see, remember the bonds present in the nucleotides. Okay, so that is glycosidic bond formed between the nitrogen bases and the first carbon atom of the pendo sugar, and this is N-glycosidic linkage is crucial for maintaining the base sugar connection. Phosphodiester bond is there, connect the phosphate group to fifth carbon atom of the sugar molecule. This bond is essential for the nucleotide formation. In addition to that, in the nucleic acids or the polynucleotide chain, we can see there is a phosphodiester link. Phosphodiester form. So here we know already there is a nucleotide is there. How can we make a polynucleotide? So there will be another nucleotide we have to make. Okay, so the pendo sugar in the fifth carbon atom, nitrogen base is there, and in the first carbon atom, nitrogen base is there, and the fifth carbon atom, phosphate group is there. So how they will link? The phosphate group will be in the second nucleotide. This is the second nucleotide. This is the first nucleotide, right? So the second nucleotide phosphate group will be attached with the third carbon atom of the first nucleotide. So phosphate group, okay, phosphoric acid is attached. When you check the phosphoric acid, here you can see one is attached to the fifth carbon atom of the second nucleotide and another one is attached with the third carbon atom of the first nucleotide, right? So there are two ester bonds there. This is one ester bond and another one. So what name we can call? Phospho. Two are there, no? So phosphodiester bond. Okay, phosphodiester bond. So remember, the nucleotides consist of only two bonds, N-glycosidic bond and a phosphoester bond. When polynucleotide chains coming, poly means many nucleotide joining with the help of phosphodiester bond. Is that clear, all of you? Yes. So remember, in the polynucleotide chains, we can see glycosidic bond, phosphoester bond, and a phosphodiester linkage. Okay. So you have to remember the examples of the nucleoside and nucleotide. MCQ, they will ask a question from this part. Okay, so adenine plus sugar gives the adenosine. Okay, so the difference, adenine is there, adenine is there, adenosine. What is the difference? Adenine is the nitrogen base. Adenosine is a nucleoside that is formed when the sugar attached with the adenine. Then there is guanosine is there. Guanine plus sugar gives guanosine. Cytidine is what? The cytosine when binds with the sugar, it is cytidine. Thymine plus sugar gives thymidine. Uracil plus sugar gives uridine. So the last four letters D-I-N-E, dine. Okay, you can easily remember because these three come under which category they are? Pyrimidines, right? They are pyrimidines. So you remember like that, pyrimidines, the last letters D-I-N-E, D-I-N-E. But the first two are adenosine, guanosine. They are the purine forms. So these are the examples of nucleoside. Okay, so let's go to nucleotides name. When adenine sugar, that is the adenosine, in that when the phosphate group, what is the phosphate group? The phosphoric acid attached, it is called adenylate acid. Okay, so this is like sugar is the ribose sugar. Okay, here adenine plus deoxyribose sugar is there. Deoxyribose sugar, then what sugar plus phosphate is there, then what name we can give? Deoxyadenylic acid. Sorry, deoxyadenylic acid. If guanine plus sugar, that is deoxy sugar plus phosphate, then we can give the name deoxyguanilic acid. Here it is guanilic acid when there is a ribose sugar. Cytosine plus sugar plus phosphate gives cytidilic acid. Thymine plus sugar plus phosphate, thymidilic acid. Uracil plus sugar plus phosphate gives uridilic acid. Okay, so this different terms, the nucleotides examples and nucleosides examples, you have to remember. Right? Then, so the last part, we are coming to the last part, some important nucleotides we have to learn. We are very familiar with this name ATP. What is another name of ATP? Can anybody say what is another name of ATP? It is some currency, we'll say. It is a currency. What is that currency? Energy currency of the cell, right? Why adenosine triphosphate is called energy currency of the cell? Because in our body, energy will be stored in the form of adenosine triphosphate. So breakdown of adenosine triphosphate, when the bond breaks, then a lot of energy will be released. There is another nucleotide cyclic adenosine monophosphate, cyclic AMP. The cyclic AMP, regarding this, you will study in that physiology part, hormones part. So, hormones, some hormones cannot enter inside the cell. For giving the message to deep inside the cell depends upon a specific molecule that is called cyclic AMP. Okay, cyclic AMP gives, it's a second messenger in cellular signaling pathway. That this part you will study more in hormones part, in the human physiology part. So remember the important nucleotides, one is ATP, another one is cyclic AMP. Then in plants we can see, not only plants, even animal cells also, you can see NAD plus and FAD. These are coenzyme nucleotides. Especially these coenzyme nucleotides are very, very essential for cellular respiration and various metabolic processes. In the respiration part, you will study about NAD, FAD. So, what is NAD? Nicotinamide adenine dinucleotide. So, this coenzyme essential in redox reaction, specifically cellular respiration. Okay, and flavin adenine dinucleotide. This is also another coenzymes required in the citric acid cycle and electron transport chain. Citric acid cycle, electron transport chain, all these we will discuss in the cellular respiration part. Okay, so which are the important nucleotides? We discussed about ATP, cyclic AMP, nicotinamide adenine dinucleotide, NAD, and FAD, flavin adenine dinucleotide. Okay, so today's session we discussed about the components of nucleotide. What are the components? There is a base, nitrogen base is there, sugar is there, and a phosphate, that together form the nucleotide. And which are the base, different nitrogen bases which are classified into purines and pyrimidines. Then we learned about the sugar differences. In DNA, what type of sugar is there? That is the deoxyribose sugar. And RNA, ribose sugar is there. Then we have learned about the different chemical bonds present in the nucleotides and the polynucleotide chain. Glycosidic bond, phosphoester bond, and phosphodiester bonds. Okay, so let's discuss some questions related to this topic. Are you ready, all of you? Ready? Yes. So let's discuss some questions. Very first one, which of the following bond is present between the sugar and the nitrogen base in a nucleotide? What is that? Answer is the glycosidic bond. What type of bond? N-glycosidic bond present between the first carbon atom of the pentose sugar with the nitrogen base. So you learned one important question, the bond present between the sugar and nitrogen base, that is glycosidic bond. Right? What is the hydrogen bond? Hydrogen bond is present between the nitrogen base pairing we learned, right? Then phosphodiester bond between the adjacent nucleotide. Adjacent nucleotides are connected by phosphodiester bond. Phosphoester bond is different, diester bond is different. Another question, in DNA, total amount of purine is equal to total amount of pyrimidine. This is known as, there is a rule called Chargaff rule. Okay, Chargaff rule that states that always purines binds with the pyrimidines. Okay, if the number of purines 10 means the number of pyrimidines also 10 only. That's what the Chargaff rule. Okay, Chargaff rule we will discuss more in the DNA part also. But here also we learned base pairing. That's why I added this question. So Chargaff rule, this is a fundamental principle, states that adenine always pairs with thymine and vice versa, guanine with the cytosine with the 3 hydrogen bond. Okay, so that this one ensuring the equal amount of purines and pyrimidines in the DNA. So the Chargaff rule is definitely explaining the structure of the DNA. How many that the number of purines equal to the number of pyrimidines. That point you have to remember. Okay. Another question: which of the following is correctly matched? Adenosine, nucleotide, thymidine, nucleoside. Adenyllic acid, nucleoside, cytidyllic acid, nucleoside. Adenosine is what? Adenosine is not nucleotide, it is a nucleoside. Adenyllic acid is what? Adenyllic acid is not nucleoside, it is nucleotide. Cytidyllic acid is also a nucleotide. But thymidine is a nucleoside. Thymidine is formed nitrogen based thymine plus sugar that form the thymidine. Okay. Next question. Nucleotide, difference between the nucleotide and nucleoside. Which component is absent in the nucleoside but present in a nucleotide? Definitely you can say it's easy question only. It is the phosphate group. Sugar is there, nitrogen base, they are both sugar bases, but phosphate group is not there in nucleoside. It is present in nucleotide. Next question. In the DNA backbone, there are different bonds: glycosidic bond, phosphodiester bond, hydrogen bond, and a disulfide bond. So the adjacent nucleotides are connected to form the DNA backbone. Okay, so sugar, phosphate, like that, the backbone will be there. Here only the nitrogen base attached. So this is the backbone of the DNA that is formed between what bond is there? That is the phosphodiester bond. Option B is the correct answer. Okay. Then next, very important competency based questions, mainly assertion reason. Nowadays CBSE and other boards and all, they added more competency based questions. Okay, especially assertion reason questions. So you have to learn how to find out the correct, how to go about this questions, assertion reason questions. Options will be given like this: both assertion and reason are true and the reason is the correct explanation, or both are true but wrong explanation. Assertion is true, reason is false, like that different options will be given. Okay, one such question related to purines and pyrimidines. So purines are double ring structure, whereas the pyrimidines are single ring structure. We already know that purines, they are the 9-membered double ring forms are there, but pyrimidine 6-membered single ring is there. Okay, but reason adenine, guanine are the purines, while cytosine, thymine, uracil are pyrimidines. Is that correct? Yes, we already remember that adenine, guanine, purines, cytosine, thymine, uracil are pyrimidines. So both assertion are, sorry, both the statements, assertion and reason, both are correct only. And reason is the correct explanation. Right? Another question. DNA contains thymine while RNA contains uracil. Is that statement correct? DNA contains thymine? Yes. But RNA contains uracil? Correct. No. RNA, the thymine is replaced by uracil. Reason, uracil is more stable than thymine. Hence it is present in RNA. It is present in RNA, but it is not stable. Which is more stable when you compare RNA and DNA? That is DNA, because of the presence of thymine. So this second statement is wrong. So assertion is true, reason is false. That is the correct option. Okay, then there is match the following. Adenosine. Adenosine is related. Adenosine is what? Adenine and adenosine. Don't get confused with these two terms. Adenosine is a nucleoside, right? It is a nucleoside. Guanyllic acid is a nucleotide. Ribose plus uracil, that form, that is the RNA base. Deoxyribose plus thymine, that is in the DNA, right? So Some common mistakes in the competitive exams and all, you may confuse with the nucleoside and the nucleotide. Remember, nucleoside base plus sugar, but nucleotide base plus sugar plus phosphate. Then the RNA DNA. Remember, RNA ribosugar and DNA deoxyribosugar. And remember the base pairing. Adenine always pairs with the thymine and the guanine with the cytosine. And the hydrogen bonds also remember. Okay, 2 hydrogen bond between adenine and thymine, 3 hydrogen bonds between guanine and the cytosine. Some mnemonics I want to tell you. So you can remember easily. I have told you one cut is what? Cut is about the pyrimidines, cytosine, uracil and thymine. Like that purines also we can say pure as gold. Purines, example A4 adenine and G4 guanine. And in between double ring. Base pairing memory. Apple tree grapes connect. Apple tree A4 adenine thymine. Grapes connect G4 guanine cytosine. Sugar difference, remember deoxy, that is missing the oxygen. Okay, so that's all. Thank you, all of you.